Good morning, everyone. Am I audible?
Yes, madam. It's audible.
So I think we have around 50 plus participants, and I think we should be starting immediately. So let me first introduce myself. I'm Rajeshri, the principal of ERNDCI Institute of Technology. ERNDCI Institute of Technology is the academic arm of CDAC Thiruvananthapuram. We are located at Trivandrum, Kerala, and we have two MTech full-time programs. MTech in VLSI embedded systems and MTech in cyber forensics and information security. So our Department of Electronics, which is conducting the MTech in VLSI and embedded system, is conducting this program, this webinar. And as you know, the webinar series is on embedded systems development and its application. And today's specific topic is instrumentation systems for life sciences research. So on behalf of ERNDCIT and CDAC Trivandrum, I welcome you all to this session. I also extend a special warm welcome to Rahul CS Nair. Rahul is a manager, technical services, instrumentation engineering, Rajiv Gandhi Center for Biotechnology. This is a central government organization, basically under the DST, right, Rahul?
Yes.
Okay. So this is again located at Trivandrum, and he has done his BTech in ECE from College of Engineering Trivandrum, and his MTech is from our institute. He is our alumni. So we are also having the pride of having one of our alumni presenting this session. So Rahul, on behalf of ERNDCI, I welcome you once again back to ERNDCI fraternity and also to this session. I hope you will also have an interesting session ahead, and all the participants here will also be having a very enriching experience. I'll leave it at that, and I hope all of you would come back for more sessions with ERNDCIT. For more information on ERNDCIT, you can come to our website, erndcit.in.
Okay, good morning all, and thank you ma'am very much for the kind introduction about me. And I also thank all the officials behind this webinar series for inviting me to this great event. And my special thanks to Divya ma'am for giving me an opportunity to participate in this great event. So before starting, let me also thank my director, Professor M. Radhakrishna Pillai sir, Rajiv Gandhi Center for Biotechnology, for giving me consent to participate in this event. So in the next few minutes, I am trying to convey a few information about instrumentation technique, which is familiar to you and that is being used by most life research people. So this life science, when you hear about life science, the main thing that come into your mind is biology, right? So life science has evolved as a field of science in ancient civilization, and at all times, research on this area is very critical and of utmost importance. So because it addresses the universal problem of existence and maintenance of life on this green planet. So like the previous century, though, 20th century, we say that it is an era of IT. So in this century, we can say that it is the century of biotechnology, or BT in short. And I work with Rajiv Gandhi Center for Biotechnology, and which is an institution under Government of India, Department of Biotechnology, and it is at Poochapura here in Trivandrum. And I work with a small department, that is a supporting department called Instrumentation Engineering Department. You can, I have the internet details of my institute in the last slide. So now let me begin. So well, you all know about the instrumentation systems, right? So whether any field, whether it is academia or research or industry platform, these instrumentation systems are all innovatable thing. So in your lab also, you are using various types of instruments, right? Yeah. Here in this topic, instrumentation systems for life science research, my humble effort is to present before you a few installations on the instrumentation side, from the very basic principles to the highest sophistication ever made to explore the very basic unit of life, which is the cell. So not only the static view of the cell and also its dynamics associated with the various intracellular activities. So I hope most of these basic concepts are, I mean, the principles are familiar to you as electronic engineers. And I also hope that my talk won't let you bored, because some concepts are already repetitions of what you have studied in your college or school days. So instrumentation systems specifically in life science research domain are aimed to probe, explore, manipulate, or to study the dynamics in one way or the other way of various cell activities, right? In order to work on an object, what is the basic essential thing is that we should see it first, right? So if you work on a cell means we should see the cell first, then we need to hold it so that we will be able to do something with it. So let me begin with an area of instrumentation which can be used to see the cell, right? So usually the cells are from 10 to 30 micrometers, and few cells are up to 200 micrometers in length or diameter, and they are mostly translucent or transparent, and most of the intracellular organs are translucent. And the human eye can detect only objects which can present some contrast information, right? So the main challenge here is to view these micro miniature structures in good contrast. So as you are aware that the basic tool to enhance this human eye for such type of vision is the microscope, right? You know, that's microscope. And this is the basic schematic of how we see, right? So light coming from the object falls through a small hole, which is about 5 mm in diameter, and then it passes through a lens. Then it passes through the inside of the eye through a jelly-like material, and finally it falls onto a screen, which is called the retina, from where the image is interpreted by the neurosignals and the object we see. So what a microscope can do is, in order to see a substance, it should reflect or refract some light. So what for minute particles which cannot be seen by the human eye, what a light microscope can do is to collect all the diffracted light coming from the object and merge it and convey it to the human eye. Here we say it as a light microscope, and so of course this light microscope has various lenses, right? Optics for providing proper magnification. So we need visible light for making these microscopes functional. But as you know, that this visible spectrum is very small in the it's a very small region in the electromagnetic spectrum, right? It is from 400 nanometer to 700 nanometer. And to be more precise, it is 0.38 micrometer to 0.74 micrometer. And when speaking about microscopes broadly, there are different classification of microscopes, but broadly, let me tell you that there are three kinds of microscopes. This is a broad categorization. You can categorize microscope in different manner also, but this is a broad categorization, because I cannot, in this topic, we cannot go to each and every branch of this microscopy in detail. So because we have to discuss more subjects. So the categories are the optical microscope, electron microscope, and scanning probe microscope. So optical microscope, you might have seen, in your in labs or somewhere you have seen. It is the earliest and the simplest of all, and various enhancements are made to this optical microscope to make it compound microscope. Different flavors of microscopes are available in this category, and you can see objects as small as 200 nanometer with this. So there are, over the years, there are several enhancements made to this. And while speaking about microscopes, two concepts are to be remembered. First one is magnification, which deals with the how many times it amplifies or it magnifies the object, and the second one is the resolution, how close two objects can be viewed. That is a resolution. That is how a microscope can resolve two adjacent points. So the next is the electron microscope, and this is also another variety of microscope which is used to see very small particles which are not possible with the light microscope or the optical microscope. And finally, the scanning probe microscope, which is beyond the scope of our discussion here. And it basically, it runs a probe, and it has a very atomic metallic tip that can move around a surface. It can roll over in a subject and can capture the image, sense the perforations in that surface, and it is incredibly powerful and can resolve subjects smaller than very, very, very small. So those are the three categories of microscopes. Also, in the coming discussion, we will come across some parts of the microscopes, right? So you may be knowing all these parts, but still I will give you a very brief introduction about this. So here you see a basic microscope, right? This microscope, in this microscope, wait. So, sorry, second. Here, so this is the any microscope requires a light source, right? Here, this is the light source, and from the here the light is going through a small aperture called a condenser diaphragm, the diameter of which you can adjust it. And from there is a stage where you put the sample. Here you put the sample here. So the stage has got an aperture like this, and there is a small clip for holding your specimen. And from the light passing through the stage will pass through then different objectives. These are the objectives, and these objectives are mounted in such a way that you can twist it to bring it to the light path. And you have different magnification like 4x, 10x, 20x, 100x. That depends on your microscope configuration. And finally, you see here, this is the eyepiece. You see here, and you can have the diopter adjustment also here, right? Depending upon your eye width, you can have, and all these are enclosed in a suitable metallic frame. With proper controls, both mechanical and electrical controls are there. Here you can see that the stage can be moved, the sample can be analyzed horizontally and vertically in XY direction by adjusting the stage, and you can have fine adjustments with this system. Then there is a base, and there you have on-off control. So these terms will repeat, and that is why I just specified it here. And the function of the condenser is to provide proper illuminating area, and it is very critical for the sharpness of the microscope, sharp image of the microscope that can produce. Now coming to the next slide. While considering the light microscope, we are very briefing about optical microscope because we have some few latest technologies to describe. So I'm not going detail into the optical microscope technology, but almost all the optical microscope will look like as before, you have seen before. So, but still for you people to understand, let me show you 4 different images from 4 different light microscope, right? These 4 different these are the 4 different light microscopes which are possible, although different configurations are there, but these are the most common. So in the first picture you see here, let me tell you. So in this picture you have seen this is called the bright field microscopy. This is a picture obtained from a bright field microscopy. As the name suggests, what it is doing is it simply illuminate the specimen and take the picture. This is the bright field microscope. And the second one, that is called the phase contrast microscope. See this one. See, you see in cells, inside of the cell is translucent. It has thicker portions, thinner portions. So when light passes through these portions, its phase changes. So special filters can be employed to capture these phase shift of various light coming from the various parts. You see, all the images are same, right? It is all the images are same, and these are the pictures of the same image seen from different microscopes. So this is a kind of microscope which records the phase contrast coming from the heat. And the third one is called it is little more complex. That is called the differential interference contrast. And here what it does is it studies the interference patterns from the samples using special optics. Then it produces the image. And the last one you see, it is the dark field microscopy. In dark field microscopy, the light is directed at a different angle. It is not directly illuminated in the specimen. And the diffracted light from the sample is focused using special only diffracted light from the specimen enters the microscope. So, and the image is constructed in that manner. So that is the last figure. And so it is basically the position or the direction of illumination. There is various combination of filter lenses to capture the phase difference or the interference pattern or scattered light that matters in capturing all these images. And each technique has distinct use in one way or other. Right. So, well, that's it for the light microscope, and we have to move on to the next technology. But before that, here are few limitations of the light microscope. The main limitation is that it's diffraction. Its diffraction limits its resolution, and also visible light is from 0.4 to 0.7, as I shown earlier. And with this, we cannot see objects smaller than 200 nanometer, right? So a theory says the resolution of light microscope cannot be smaller than half of the wavelength of visible light. So visible light starts from 400 nanometer. So half the wavelength is 200 nanometers. So that is the maximum size that can be viewed using light microscope. And the internal structure of the cells, like you see here, are colorless. Now you can see only the brightness or gray-white variations. These are all colorless and transparent. So to increase the contrast, to increase the contrast and to provide coloring, which in life science researchers term, they use what is called the labeling, right? Colored labels. It can be given to these images, and there are techniques for this. And also, sometimes these people want to study the motion of the cell, right? Living cells, they want to study the intracellular motion, interaction of different cells in tissues. And these things are not possible in light microscopy. But colored images can be obtained using by staining suitably, but still there are limitations. You have to use different stains, and the number of stains are limited. And also contrast is also limited. So the solution, we have more efficient techniques for imaging with color and contrast that we will discuss next. So that is the fluorescence microscopy. And fluorescence, right? Fluorescence is a property of particular material. When light falls on it, what happens is that it emits a different light with different wavelength, and that is a property of fluorescence. And here there are special chemical compounds which can fluoresce accordingly, and we attach these different colored compounds to different parts of the cell by using suitable cell preparation procedures. And then we will place it before the fluorescent microscope so that the fluorescent microscope can give you, I will show you beautiful images from fluorescent microscope, right? So the main ability is that the internal structure of the cell can be viewed, unlike the other image, previous image we have shown. So it allows colored labeling, and it has more 1 to 2 times resolving resolving power than a light microscope. And it also allows to track the internal dynamics of the molecules. That is, live cell imaging is possible. And that too, in color, you can see the time-lapse image or video of movement of cells or movement of cells within a liquid or tissue or whatever. Now, some advanced concepts associated with fluorescent microscopy. It's a very broad subject. In fact, I am not uh, competent enough to get into that. Fluorescence correlation spectroscopy and fluorescence cross correlation spectroscopy are such type of things. These two things we won't discuss it here, and just for the information, I am presenting. Now see the beautiful image. This is the beautiful image from the fluorescent microscope. You can see the cell. It's more clear. Now you can compare it with the other image, right? So various internal structures are visible and in different colors. So let us see how we can explore the cell in this fashion. Okay. So fluorescence I already said, and substances which emit fluorescence are called fluorophores. And this fluorescence microscopy requires extensive sample preparation. So you need to select proper dyes, fluorescent dyes, and then you need to do some specific lab work so that these fluorescence dyes can be attached with different internal organs like nucleus or cytoplasmic parts or ribosomes. Those are the things inside the cell you might have heard in your old school days. So that is a basic thing about the fluorescent microscopy, and it principally is same. You know, as electronic engineers, you are all aware, you are all familiar about this diagram, right? This is the Jablonski diagram. So when a fluorophore is hit with a light of particular wavelength, suppose excited with an excitation light, its electrons get emitted to an excited state, and then it comes from to the ground state and releases photon, right? So when we excite, see the diagram, when we excite it with a particular wavelength, emission occurs slightly shifted from the excitation. And basically the difference is the stock shift. You all know about all these things, right? So this is the basic principle of fluorescent microscopy. So in fluorescent microscopy, what we use is we attach different fluorophores with the internal, with the subject of study, and then we make the sample suitable to present before the microscope, and the microscope can capture the image of by sensing different excitations from different parts of the cell. So detailed. This is the typical arrangement for a fluorescent microscope, and here you can see we have a light source here, and the light from the light source, we put a special filter, you know, because our fluorescent dye may be sensitive to here green colored light is showing. So this filter filters only the green light, and whatever the dye is used with the sample is also sensitive to that particular wavelength. And it is focused on to the specimen, and from the specimen the emitted light, you know, emitted light is directly coming to the eyepiece or the sensor. Here, this is the piece, the optics, you know, it is called a dichroic mirror. The property of the dichroic mirror, on the right side, you can see a filter cube, and this is called a filter cube. It is an actual view of the filter cube. And you can selectively pass light through a filter cube, right? So the excitation light can flow like that, and it cannot go backward to the eyepiece or to the camera. And here only the excitation wavelength. So in order to further filter the emitted wavelength, we put one more emission filter here before sensing or before viewing, and it is the emission filter. And finally we see the image, and we will explore how we will see the image. Okay, we will explore one by one, and this is the basic principle of working of a fluorescent microscope. And to a more detail, see, this is the basic arrangement. Like the earlier light microscope, you know, all these optics are fixed onto a suitable this frame here. Which so this is the frame, and we have the light source here, right? Here is the light source, this small dark colored part. So you can see the light coming through this, and various optics. It is guided by various optics here, and then the light dichroic mirrors are equipped here, and it falls to the sample. Of course, through your suitable objective, you can twist it to the correct direction. Then the emitted light again goes back above. You can see it as red, and here is the CCD camera, and the you can also view it through the eyepiece. Almost all the microscope has different optical ports through which you can capture the image, or you can see the image. So there are ports with 100% transmittance, 80% transmittance, like that. You can attach multiple cameras with to get different images, different images of interest. So this is the arrangement basically. Here, some of the important main three parts are there: the optical system, then the light source, and the detector. So we will be concentrating further on these three important concepts for a fluorescent microscope. Now, to explore the more detail into the filters, you see the filter cube. You can see the filter cube. You know? This is the filter cube I presented earlier, and it has an emission characteristics also on the right side. You can see that the emission light, you know, red light, sorry, excitation light is band limited, right? You can see the red curve, but the emission filter, it passes. That is a characteristics of the filter, and these filter cubes are mounted in a special assembly like this on the microscope, so that you can see the teeth on the surface. This is micro positioners or stepper motors are moving these filters. There is no, even though you can control it manually, usually these type of microscopes are driven from software. So there are also linear line filters like this, and you can push it or pull it back so that the appropriate filter comes into the light path. So this is the overall looking. Here is how it looked like. This is from Leica's Microsystems. A simple fluorescent microscope will look like this. It has got an attached CPU, then camera, then binocular, then touch panel for adjusting various objectives or various selections. You can put camera here or camera there. So different ports are also available. Now the stages, you know, various parts like mechanical stages are all computer controlled actually. These stages can be manually adjusted, but still for fine movements, it requires the use of a CPU. You can see one type stage. For just for demonstration, let me show you the movement of a particular XY stage. It moves like this with precise positioning systems. And there are joystick-like controls for positioning for adjusting these stages. These stages can be procured in module-wise. So that's the stage, and here another demonstration of a C stage, because it moves in the Y direction. Actually, this is not the stage that is used in the microscope, but this is the image I got when I searched on the Google. So it provides a movement in the Z direction. So these are all the motorized stages. So like I said, there are three main parts: the light source and optics and detector. So you have already seen the optics. So main light source with microscope is mercury arc lamp. So mercury arc lamp means it is a very small thing to see, but it is highly sophisticated because it is manufactured from quartz under high pressure filled with mercury. So it should be handled in accordingly, and there are separate power packs available to power these uh, uh, light sources. So other than mercury, there are xenon arc lamp is also there, and you can see the mercury arc lamp spectrum here. It covers almost the visible light range from 400 to 700 nanometer. Intermediately, some lights are missing somewhere, and that is the main disadvantages of this mercury. So usually these mercury lamps are operated with a timer because it has 200 hours of lifetime. So we need to know how much time has expired. So the power pack usually comes with a time built in, and almost it gives you a intensity overall spectral emission from all over the light visible spectrum, right? And then next is the
Hello, Rahul. I think the slide has gone smaller now.
oh.
I think someone is trying to present now. That's why.
Oh, shall I
So,
stop and start?
go to the presentation slide once more. Stop and start once again.
One second, one second. One second, ma'am.
Yes. And participants, all of you, please mute your audio and video, which will interrupt the section.
Shall I present it again?
Yes. And participants, please mute your audio and video. Yes, Rahul, you can put it in a slideshow now. Yes, now it's okay now.
So it is. Shall I proceed?
Yes, okay.
One second, ma'am. So this is a spectrum of xenon arc lamp. So as compared to the earlier one, that is mercury arc lamp, it provides much better visible spectrum of light from especially in the visible region. So these are the two common light sources that we are using. And apart from this, sometimes tungsten sources are also used. And tungsten is also a very good light source. Now, the most advanced microscopic techniques use lasers, because lasers are very crispy and it's very highly coherent. And you need to use different lasers for different spectra, because that is the only disadvantage. You need to use a green laser, red laser, like that. And also it is costly as compared with the other light sources, and you see the sharp emission spectra from lasers. Now recently there are solid state lighting is also available, which are the LEDs. So it is usually called light engines. These are much costlier, and with these devices have integrated array of powerful LED sources, and they have inbuilt optical filters. Usually the light is guided from using a optical fiber or a liquid light gate. So that's all about the light source, and then comes the detector part. So the detector is usually the CCD camera. The CCD cameras are of different nature. There are CCD cameras and EMCCD cameras. So depending upon the applications, the end user can change the camera accordingly. So compared to CCD camera, the EMCCD camera, which is the electron multiplying type CCD, that is EMCCD, provides much better magnification, much better clarity. The documentation says like that, but even though the background noise is less compared to the CCD, the documentation says it is somewhat the multiplication noise associated with the processing of the signals is more projected onto the images from these cameras. Anyway, these cameras are highly costlier cameras, and cooled CCD is used with Peltier technology for cooling. And this camera cost, it seems, a 2-bedroom flat in Trivandrum city, this camera alone. So there are other enhancements made to this camera, and one such thing is the CMOS, that is a scientific CMOS, and documentation says the CMOS is of higher frame rate and very comparatively very lower noise and higher dynamic range also. So all these setup, the microscope setup is needs to be placed in a vibration-free environment, right? So for this, it should be placed in a specially anti-vibration, fabricated anti-vibration table. And you can see here, it's a heavy platform, nearly weighing 110, and it is placed in bellows. And here you can see a pneumatic valve here, and these bellows are all inflated with suitable air pressure, so that if something happens to some disturbance occurs, it will automatically maintained by the mechanical level sensors fitted to these 4 legs. So it provides a vibration-free set functioning of the microscope. And I will show you. Sorry. So that is it. That is it for the fluorescent microscope. Now we move on to a next type of microscope, which is more, which provides more sharper images than the fluorescent microscope. And here you can see, two images on the top. Here you can see 2 images. See, this is the image that we obtained from a fluorescent microscope. So, but this is somewhat gauzy, right? We cannot see much more clear, because the other part you see, it's much more clear, right? More clear. In the bottom portion also, the same image with the 2 microscopes you see. On the left hand side it is more clear, on the right hand side it is not much clear. So this blurring is mainly because of the scattering of light. So we need some technology to avoid the scattering and capture only those particular emitted light from specific area. So so let us see how we will achieve this. And the microscope used for this, you can see the title, it is confocal technology. And this is the basic principle of the confocal technology as compared with the other one. The main difference is that the optics, everything is arrangement, everything is same. But here the main difference is that they will put one pinhole here, right? They will put on pinhole here, so that selectively it can pass the light, very small, it is a very small aperture. So the excitation light comes to it, illuminate the surface, and again emitted light comes through the dichroic mirror. Again there is on pinhole, so that only those portions. Hello? Hello?
Hello. No. It's audible.
Okay. So the emitted light from the specimen falls onto the detector. Here you can see there are 2 emitted light spectra on the light out of focus light specified, with this pinhole. These out of focus light are completely obstructed, and only in focus light enters to the detector, and that is the reason for the crispy image. And as you see in the image here, you can see here in this portion, you can see this crispness is mainly because only the in focus light is captured because of the pinhole. So we have pinhole both at the input excitation side and at the emission side. And this side, on the right side, more simple representation. See, the violet rays are the source light and it is focused on the image. And suppose the out of focus light from a different plane, without this pinhole, it will also come into the detector. But this pinhole limits these rays, and only the in focus light labeled green will enter into the detector. So there is a basic arrangement. So to a more clear view, this is the pinhole and everything, and this is the light source. Let me take the pointer. So this is the light source, and this is the pinhole. Through this pinhole, it passes through the excitation filter, and finally it is focused onto the sample, and then the excitation light, emitted light is captured. Only the focus light is captured to the detector. Here the detector is a photomultiplier detector, or a PMT in short, because these cells, the emitted light from these cells is very, 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 very feeble. So we needed to use some kind of amplification before it is fed into the camera or naked eye. So that is why it is being used. And you can see the pinhole here. And here also the pinhole is there. And another important thing to be noted is that you can see 3 focal planes here, right? 3 focal planes. So this is the speciality of confocal microscope. Suppose with a given sample, you can, by adjusting the optical system, or by adjusting the intensity of the laser or selection of lasers, we can focus on different layers within the sample. And we can focus different layers, and we can scan this each focal plane in a row by column matrix manner called a raster. So it is the raster screening. So there is a special feature of the confocal microscope. So this you can, with a given sample specimen like this, you can capture each focal planes individually, and then the software, by using specialized software which is developed for this purpose, you can construct the 3D image. Sorry. The 3D image. So these are the different focal planes. These are the different focal planes. And the software can construct the 3D image of the of the specimen, and with sophisticated software tools, you can turn this upside down or tilt it, or whatever you can do with this. And this is another setup that should be mentioned. See, I told you regarding the raster scanning, right? This raster scanning means actually the laser light coming in this way, like this. There are X-ray scanners, which are lenses, which are basically mounted on special motors called galvanomotors, which you may be aware of. And these motors can precisely position the beam, like, see, the raster scanning. So this is the raster scanning arrangement. And that is the 3D reconstruction from the microscope. And there are 2 types of confocal microscope. The one type for is called a spinning disc. It's called a spinning disc. It will look like this. This is only the scanner part. Here you can see the spinning disc. You can see the spinning disc here. So this is alternative to the galvanomotors that we saw earlier. So the spinning disc will guide the laser for scanning. And the other arrangements I have already mentioned. These are the filter sets, and this is the light path, and this is the camera port, right? So another arrangement is the confocal that we have already discussed. This is the raster scanning type confocal unit. So like I said, the laser is coming through this point. This is the input port, and from there some kind of intensity or adjustment is there. Then dichroic mirror deflected to this for excitation, and there are different filters: excitation filter and emission filter. Then there are spin hole turret, then various types of mirrors and galvanic motors for position control. So this entire unit will come as a box that can be placed near to the microscope. Typically, a microscope will look like this. The confocal microscope will look like this. See the basic microscope here. This is the basic microscope, and this is the scan head, and various optics are here. And you need a powerful computer for this purpose, usually with much wider screen, because almost all the operations are automated through software. So you need a separate monitor for controlling the software. So adjusting the controls, various controls like stage height, then selection of wavelength, selection of excitation, emission filters, then movement of camera, something like that. Then you need an extra screen for viewing the image alone. So there is a conventional arrangement of a microscope, and let me show you beautiful images from this confocal. These are some of the few images, and to be frank, I don't know the details of these cells. My colleague here, and I told you that it is possible. Live cell imaging is possible with confocal microscope or fluorescent microscope. Here is a moment recorded, a time-lapse image of video of what is recorded here, and you can see something disappears from the picture. So these are very important points for as far as a life science researcher is concerned. And there is one more image. These fluorescent dyes, see, some of them are popping up and some of them are disappearing. So likewise, we can record very minute cell dynamics with microscope. So in short, we can say that this this confocal microscope can provide a sharp image, right? Sharp image, and with less background noise, and it has an ability to control the depth of field also, and 3D image recognition, everything is possible. And one more subject to be specified is the perfect focal systems. For live cell imaging, sometimes you need to focus the sample over nights, okay? Overnights. In such cases, suppose what happens if a mechanical drift occurs. So there are mechanisms which are provided within the microscope which locks the objective to the particular focus of interest, right? You can see this. A separate light is used, light source is used for this. This is usually 820 or 800 nanometer wavelength, which is a higher side of the visible spectrum. And it always keeps these objectives in perfect shape. This mechanism measures the correct distance here. So there are motorized mechanical parts that can adjust these objective to the, even if a drift occurs, this closed loop system will work in such a way that this entire mechanism will come into focus, perfect focus. So that is a perfect focal system. If you have a more detailed image of this, this is the perfect focal system assembly here. So you can see this red light, right? This red light is the light used for focusing alone, and it is not mingling with the imaging light. So that is the difference. The light sources are differently placed. This is the LED for this thing, and this is the light source for imaging. So that is the perfect focal system. So this is our, all these setups are in our institute, and this is the typical view of a confocal microscope as seen from our institute facility. So you can see the various lasers are stacked here, and the last box is the central processing unit. Various lasers are stacked here, and the microscope is placed in a vibration-free table. And this is the pneumatic setup for the vibration-free table. And And other than this electronics, there are electronics on the other side also. All these are kept in a closed transparent sheet-like material, which is called the incubator. And the incubator is actually, during live cell imaging, you need to mimic the living atmosphere, because your body conditions you need to mimic. Suppose if you are scanning human cells, you need to mimic, that is 37 degrees centigrade with 5% CO2, because your respiratory system already maintains oxygen in your this thing. So we cannot keep it open. So we needed to maintain the CO2 level also. So 37 degree and 5% CO2 condition we will maintain in this chamber for live cell imaging, and for overnight imaging is possible with this. And you can see cylinders for this, gas cylinders for this, which is supplying proper gases for maintaining the environment. So next comes other microscope, which is the, suppose if you want to see much smaller details, which are 100 nanometer or 50 nanometer thickness, what will you do? Then you need a light source, which is much sharper than the light source. You see this image here. You are familiar with this image, right? These days. So how to see a virus like coronavirus, you know, it is only possible with light waves, which are shorter than, waves which are shorter than light waves. So usually electron microscopy deals with electron waves, which are nearly 0.2 nanometer in wavelength, which can vary up to 0.2 nanometer. So these type of viruses can be excavated using the light microscope. You can see the scale of resolution for various microscope. For electron microscope, it is very deep down. You can see even small proteins, lipids, or small molecules. So that is, here is the setup. There are two types of electron microscopy systems. Scanning electron microscope, and the scanning electron microscope is something that can give a 3D image. And transmission electron microscope, the picture shows one such transmission electron microscope in our facility. So when you say that electron microscope, this the word micro is only in the word, because it is very bulky instrument. So these are the vacuum systems and other part. I will show you the details. There is a electron source, and it is focused using electromagnetic means. Electromagnetic focusing is used, and the sample is inserted in between, here somewhere in between. Here you can see the sample port, and you can view, unlike the other microscope monitors here, phosphor screen is used to view the electron microscope. Afterwards, it is captured to a suitable camera mounted here in the camera port, and it can be captured to a computer for further processing also. See the binocular. If you want to see the phosphor screen, it is closed now actually. So if you want to see the binocular screen, you can use the binocular also, and you can use these controls to adjust the high tension voltage or panning of this area of interest. So that is the electron microscope. Now we will move on to another subject. So suppose in some situations we need to find out the concentration of unknown solutions. So determination of impurities, or quantitative determination of some compounds, or some functional groups, or we want to understand how much range of wavelength a particular substance will absorb, which is termed as kinetic studies. So in those areas, we use what is called the UV bar visible spectroscopy. So spectroscopy is basically the interaction of light with matter, and here is the simple arrangement for this instrument. You see there is a small light source, which is usually a tungsten lamp with necessary filters, which can be projected, the particular wavelength can be selected by means of a monochromator. And there is a beam splitter. We have a reference sample. So the reference sample is also already a standard sample, and we know the concentration. So we know the absorbance value. So if you put an unknown sample, we will get some reading. So by correlating this reading with the reference sample or the curve, we can detect the absorbance. By detecting the absorbance, we can detect what the concentration is there in the unknown sample, right? There is a formula for the absorbance. You are familiar with this formula, and this can be further tuned to like this, A into epsilon CL. This is the Beers-Lambert law actually. You're all familiar with that. So this device operates on that principle, and Here is how it look like. So UV visible spectrometer will look like this, and these are the cuvettes. In the earlier formula, you see that ECL. L is the path length. So these cuvettes are of different. This is 10 mm cuvette like that, 0.5 mm. Sorry, 5 mm cuvette like that. So different cuvettes are there to hold the sample, and the instrument, the reading can be obtained from the instrument itself, or you can plot it by attaching a computer. So that is UV visible spectroscopy. And another instrument of this kind is the IR spectroscopy. It is almost also same, but it is used to, it is little more complex. It uses IR source, which is a longer wavelength than the V range. And it is also called vibrational spectroscopy because it studies relation between the bonds. It mainly, it can be used to identify the structural groups present in a molecule. So like that only they are telling. I don't know the much details about this. And you can see the graph on the right side. This is information because suppose if a particular bond has a molecule has a double bond, and that double bond has a vibrational motion of 1,000 hertz. Then if you excite that molecule with 1,000 hertz, means that 1,000 hertz will be absorbed. That is the basic principle. So in the graph, you see the wave number on the top y-axis, you see the transmittance. So if a particular wave with a particular 1,000 means, here you see 1,000, 3,000 means that vibrational motion or elongation motion of that particular bond is 3,000. That is why it is absorbed here, and you can see a depth in this. So likewise, we can study the presence by comparing it with a reference and unknown spectra. So this is how an IR spectrometer looked like. Even though I just described it, it is quite complex and requires one separate session. Then another thing is the mass spectroscopy, right? Mass spectroscopy, as the name suggests, it is used to find the molecular masses. This is quite complex equipment. Unlike the other two spectrophotometers, this is a quite complex equipment. And so different techniques are available to study the mass, NMR, X-ray. But here we use, but mass spectrometry is the more accurate for studying the mass. And it can explore compounds as even to the, all the independent components can be studied. So here we are concentrating only on mass spectrometry because we have in our facility, there is a mass spectrometer here. Usually proteomic studies are done with mass spectrometry. So basic arrangement for this is as shown in the figure. So in order to analyze a sample, you inject the sample to the sample port, right? So the sample gets heated so that it gets vaporized to a gaseous form. And in the meantime, you need to hit with highly accelerated electrons so that these molecules decomposes into their component parts. Then only we can study what are the parts, what are each components present in it, like that. And then these molecules are accelerated by means of external magnetic field to a detector, where the heaviest molecule will reach only lately, lighter molecules, because this deflection is mainly to sort out the neutral molecules, neutral ions, and only to pass those ions which are interested to study, which are charged. So the lighter, through the length of the tube, these lighter molecules will reach first. So accordingly, the machine is calibrated. So heavier molecule will come late only. See the spectra down here. You can see mass intensity 29, intensity 29 initial, but intensity 98 comes late. So this is the mass per charge ratio, and this is a typical graph that we get from mass spectrometry. And there are two types here. There is a matrix assisted laser desorption or ionization type mass spectrometry and electrospray ionization technique. Both these are here available in our institute. In the first case, laser is used. So in order to make the sample sensitive to laser desorption, it is to be prepared. Suitable sample processing techniques are used for that. Matrix is actually a chemical compound which can be added to a sample in a very huge ratio. For one sample, 10,000 metrics are added, so that sample won't get deteriorated. And so you can study it with the laser. And in electron spray ionization technique, you cover the sample with a particular buffer molecule and then heat it so that the solvent escapes. And then we analyze the ions. So it is basically the difference lies in the method to produce ions from the molecule that is being analyzed. This is the setup with our instrumentation system. So you can see this white portion. This is the Waters HD 5000 machine, and this is the injector portion. All other systems are basically meant to introduce the sample into the unit, and this is the work surface for this. And you need hydrogen, nitrogen, argon, similar gases for at different various stages for its operations. So these are supplied through proper gas regulators. So this is the entire setup of the mass. And if you see these instruments closely, you have robotic cams. You can place the sample here, in racks, in small sample racks. You can place it here, and robotic cams will pick it up and analyze the sample one by one. And after analysis, it will place these samples in different. So it is very complex machinery. You can see stepper motor assembly for manipulating the samples, movement of samples. And this is the mass spectrometer. That is, the other one is the ESC system. Here is the MALDI system. So the length is because it is a time of flight tube, larger. See, these ions has to fly a distance before it reaches the detector. So you need a tube, vacuum tube. All these instruments are under high vacuum only. So that is the MALDI setup. And another piece of equipment is the chromatograph. So chromatograph means you can separate components from a liquid mixture. There are various chromatograph, which is a gas chromatograph, liquid chromatograph, and ion exchange. But we use here we discuss only liquid chromatograph here. And while discussing the chromatograph, two important terms are to be worth mentioning. The first one is the mobile phase, and the second one. So mobile phase and a stationary phase. You can see in the bottle, there is some mobile phase. Means for a gas chromatograph, it is a gas. For a liquid chromatograph, it is a liquid. So you need something to carry your sample throughout the instrument. I will show you the setup. So which is for that purpose, this mobile phase is used. And separation of components occurs in a stationary phase, which is called column. That is on the right side here. These are steel columns which are packed with special materials depending on the application. And this is a typical setup of HPLC. So you have the sample here. You inject the sample to the system. You have the mobile phase here. So the pumps are constantly pumping the liquid. So along with the flow, you inject the sample. So this liquid will carry the sample through the entire system, right? So the separation occurs onto the HPLC column. So this column is packed with different materials, maybe silica or something of that kind, where the separation occurs. So depending upon the particle sizes, these are bonded to different parts of this column packing. Finally, they are eluted by means of by pumping out a buffer solution, and detector detects and provides a peak. You can see a diagram of this. As far as this LC is concerned, elution time, in the x-axis it is minutes. So different retention time, different elutions comes. So with the non-compound, if you have a diagram like this, if you are analyzing a particular peak somewhere here, then you can identify that this is a particular thing. And also you can quantify this curve area. Everything is also important. So that is the setup. Here in our institute, this is the setup. You can see the various pumps. These are pumps, and these pumps are very sophisticated. It can very accurate also. You can pump even to microliters per minute. Think of a pump that is pumping microliter per minute. So these are that much accurate. So HPLC usually works in that range also. It's pumping. It's very accurate. And then only you can capture each component individually. Then this is the column oven. Column, we cannot place the column at outside. It requires a special temperature control because elution depends on temperature also. So this column usually ranges from 40 to 60 degrees centigrade. You can adjust the temperature. That depends on the protocol of the experiment. Then you can have a number of detectors here. You can have an electrochemical detector, or you can have a photodiode array detector. several, and you have a computer for interpreting the graph. So this is the setup that we are using here. So another piece of equipment. So why I am making it fast is because we have no time. So another piece of equipment is the fluorescent assisted cell sorting. So cell sorting, if you have a mixture of cells of varying sizes, you can have cells of varying sizes and you can have wells of varying sizes and with various complexities. Complexities means internal details. So that we can use it with the facts. Its full form is fluorescence assisted cell sorting. So it is basically a technology called flow cytometry. So while discussing this, one thing that is worth to be mentioned is the hydrodynamic focusing. So hydrodynamic focusing means that you can focus the sample or the solution containing the cells to be analyzed by means of a fluid, a special fluid called a sheath fluid. So the sample jet is introduced through the middle of the sheath fluid so that the sample stream remain intact and it can be analyzed. Each cell can be analyzed individually. I can show you the details of it. See, this is the sample stream and it comes through a nozzle of 0.5 micrometer diameter. So each cell, this is the dark colored portion is the sample containing different cells. So, and the outer portion, light blue, is the sheath. So by hydrodynamic focusing, this jet stream passes through a liquid stream at the center and it can be illuminated with a laser light, and then we can study the diffraction or scattering either forward or backward like that. So you have different pressure setups for the sample and the sheath fluid for hydrodynamic focusing. Accordingly, we will do this. So this is the concept. This is the nozzle, conical portion, and this is the sheath fluid. And this is the central core through which the sample comes, and at the nozzle, see, one by one cells are coming. So we can study the cell one by one. I will show you how these cells are analyzed. So basically, this is the setup of the facts. There is a laser beam coming from this, and there are several optics. So usually a straight detectors used are photomultiplier tubes because very feeble light from these cells. So they are finally collected to the ADC and is processed. So this is the basic thing. The core portion is this nozzle, that is the hydrodynamic focusing. So here we can mechanically, we can physically sort the cell also. Cells of different sizes can be sorted out. So this is a typical diagram. You can see dot plots with population, cells of particular size. This much of cells of particular size here, this much of cells of particular size. I will show you in detail. So here is the simple animation which you can finally see. The solution contains a lot of cells, with complexity. See the internal structure. There are different cell sizes. The internal structure also differs, and all these cells can be put into this in a tube, and it can be fed to the instrument for measurement. And this will give you a graph, the one that is shown here on the right side. So the sample can be loaded accordingly. So there are various optics with filters that you can detect. Detectors with different detectors are used with different wavelength of light. So you can analyze each cell independently for its physical size or internal complexity. I will show you. You can give the details here. Once the sample is loaded, the instrument sucks it up, and it is presented by hydrodynamic focusing before the laser. So once it is placed before the laser, means laser light goes forward and sideways also. This is called forward scattering, and this is the side scattering. So forward scattering actually gives a picture of the size of the cell, as you see here. See, when a cell comes at the light, see, forward scattered light, you can see the size of the cell. It is actually the size of the cell that matters, and you can use deflection plates with the electric field to sort out these cells to specific tubes if you want. Now there is another more scattering, that is side scattering. The side scattering depends on the internal complexity of the cell. The cells contain various, here for different shapes, different forward scatter, different cell sizes, right? Similarly, in the case of side scatter, so this forward, depending on the forward scattered light, this detector will produce peaks accordingly, depending on the size. So it can be plotted with the forward scatter information on the x-axis and cell count on the y-axis. And this is the diagram that is typically from obtaining from a fax machine. And you can also study the side scatter. In that case, use a separate optics to study the side scatter. See, depending on the internal complexity, the side scatter varies. This can also be detected by means of a detector, and it can be suitably recorded. Each pulse will be depending on the internal complexity is less, pulse will be less, proportional voltage will be available. So, actually, so this is the another histogram showing that recording. And you can combine both recording, that is forward scatter and side scatter, to obtain dot plots, so that cells of particular population, that is a particular size and complexity, are grouped like this. So you can sort out this also. This is the basic technique that is used in this. Now this is the fact setup. Here is the basic instrument, and it has got a fluidics card for the sheet fluid for dynamic focusing, hydrodynamic focusing. It is kept beneath, and these are servers. So this is a setup here. So, and another equipment. Now coming to another equipment, this is a PCR machine, and you might have heard this word recently because most of the newspapers are specifying PCR facilities here, PCR facilities there associated. Well, this is a machine that is well suited to find, analyze viruses, or to find the presence of a virus. So PCR actually stands for polymerase chain reaction. So polymerase chain reaction is basically for amplification of DNA. Suppose if you have a small DNA component, DNA is a very important molecule as far as this life science research people are concerned. So if you want to identify a particular virus, say coronavirus, means you want to identify the know whether the DNA of that particular is present in your body or not. So for that, they will take samples from your body, they will analyze that, they will look for the DNA of that particular. That is a basic technique for PCR machine. So this is a PCR process actually. Here, with single strand of DNA, through these processes, we can have multiple copies like this. Finally, we have multiple copies. So basically, what this used is, it heat the sample to, see, in step 1 to 95 degree, then it reduced to 55 degree, then further to heated to 72 degree. So the cycle repeats again. Each cycle produces fixed number of copies. So the PCR machine does this job. This is a machine. We have thermal, these are called Peltier operated thermal blocks. So this is the close view diagram of this. It actually uses a thermoelectric Peltier module for heating. And this is a PCR plate where various samples are loaded. This is a typical PCR machine. This differs from the earlier one. It can do condensation also. See here the arrangement. The plates can be loaded. Simultaneously, we can label the sample with dyes as set in the fluorescent microscopy, and we can view the emission spectra. See, emission spectra, and by comparing the spectra, we can know whether a particular virus is present or not. Then comes the DNA analyzer. So DNA molecules actually, it consists of a sequence of small particle bonding in unique pattern. It is unique for each and every individual, each and every organism in this universe. So if you want to read your particular DNA pattern, you can use this machine. So there are highly advanced technologies for getting the DNA sequences. You see right side, this is a chip with the wells on it. You can prepare the sample, load this chip into this chip directly, and the machine will give you sequencing after a series of run. It will give you the sequence of the CNA. This is actually, both these machines are with our institute. 1 second. So that is a DNA sequencing machine. And suppose there are systems where there is no human intervention is required for sample preparation. You see, these are all robotic arms that can do, once programmed, they can pick up the sample, they can aspirate the sample, they can do whatever mixing or PCR, everything they can do, and only you need to go and take the result. Just load the sample and take the result. So there are systems like liquid handling systems available. Besides all these things, wide variety of instrumentations, minor instrumentation set up from small pipettes, electronic pipettes that can accurately pipette the solution below 1 microliter, as low as 1 microliter, then to incubators of various kinds, shaking platforms of various kinds. Then for cell storage, we have freezers, ultra deep freezers, cryo rooms. A lot of instruments are all there. So I hope that's for it now. And this is our institute, a bird's eye view of the institute. And I needed to acknowledge few persons. Rajshekaran is our AGM, and Sajan is a colleague of mine. Dinesh is also working with us. So you can have more details about our institute at this domain, that is rjcp.res.in. And you should send me that, because the presentation was very fast because I don't have enough time. So I hope this was not a boring session to you, and you should write feedback to me, and suggestions are also welcome. So this is my email address. Hello, ma'am.
Okay, Rahul. So it was a good session. So participants, if there are any queries, you can post it in the chat.
So shall I go to the... Hello.
Hello. I think the session was explained deeply and in a good, clear manner. So I think we can conclude the session, and I request Mr. Kader, Principal Engineer of Here and DC80, to say the vote of thanks. Sir, please.
Hello, Kader sir. Hope you
Good
remember me.
afternoon. It was a very informative session, and we are really inspired to have Rahul to give us his experiences about the different information systems that he has been working on. And thank you, Rahul, for being part of our webinar series. And we hope that you would come to us in future also to give us your experiences, and hope that your research is of great fruitfulness for the what you call detection of these kinds of viruses and all those things. And let it be really really fruitful. And I wish you all the success in your future endeavors. And thank you all participants for attending this webinar. And we hope that you would attend the forthcoming webinars also. And we are really happy to be associated with all of our students and from all of us here at RNDC IT, and from students and lecturers. I wish to thank Rahul for taking the time out to give a wonderful presentation. Thank you, Rahul.
Thank you.
Thank you, Rahul, and thank you all the participants. And we can end the section now. Thank you all.
Thanks for watching. Drop a like, leave a comment, and don't forget to subscribe.